Biochemical Basics of Essential Amino Acids. Part 1. Proteins and peptides are at the center of all living organisms. At the core of our entire biochemistry, we find proteins and peptides, which are the essential building blocks of macromolecules and vital for a multitude of biochemical processes. Amino acids, more specifically L-alpha amino acids, are the smallest molecular unit of proteins. Amino acids are not only essential for the preservation of our muscular tissue, they are also vital for the production of enzymes, hormones, and neurotransmitters in the brain. Furthermore, they can also be used as a source of energy after other sources like carbohydrates and fat are not available in sufficient quantities. The process in which our body converts amino acids into glucose is called gluconeogenesis. The 20 amino acids directly encoded by our genetic code can be categorized in three major groups. 1. Essential alpha amino acids. 2. Conditionally essential alpha amino acids. 3. Non-essential alpha amino acids. Our body can produce most of these amino acids from scratch through biosynthesis. These amino acids belong to the class of non-essential amino acids. However, there are 9 unique essential or indispensable amino acids which we have to supply to our body with our nutrition because we do not have a biochemical process to build them from other amino acids or smaller molecular components in our body. These essential amino acids are critical for the production of countless proteins and peptides, which are crucial for all kinds of life-supporting biochemical processes and the preservation of our muscular and skeletal systems. About 50% of the amino acids coming from protein in our food are used to produce enzymes and non-muscle related biochemical and physiological functions. Besides the function as a basic building block for proteins and peptides, amino acids have also non-protein roles in our organism, and they have important functions as metabolic intermediates in the biosynthesis of important neurotransmitters and other biologically highly effective compounds. Some examples of amino acids as biological precursors are worth mentioning here in this context. Tryptophan, for example, is a precursor to serotonin, a very important neurotransmitter in our brain. Alanine is an important source of energy for our muscle and boosts the immune system by producing antibodies. Arginine is essential for the wound healing process. Glutamic acid is a precursor to glutamine and GABA. GABA standing for gamma-aminobutyric acid, which plays a key role in the disease of epilepsy. Glycine and glutamine are precursors of nucleotides. The failure to get enough of these amino acids has serious health implications and consequences. A deficit in essential amino acids will ultimately lead to the degradation of our body proteins and loss of muscle mass and dysregulations in multiple biochemical reactions. The 9 indispensable essential amino acids and the minimum daily quantities are listed in a WHO publication called Protein and Amino Acid Requirements in Human Nutrition. The World Health Organization makes the following estimates for the 9 essential amino acids. Histidine, 10 milligrams per kilogram body weight per day. Isoleucine, 20. Leucine, 39. Lysine, 30. Methionine, 10. Cysteine, 4. Phenylalanine and tyrosine, 25. Threonine, 15. Tryptophan 4, valine 26. All these indispensable amino acids are in our food. However, we can spot significant variations in the qualitative as well as quantitative composition in our nutrition. The amino acid profile or composition and the bioavailability of proteins in our food are critical for the smooth operation of all our biochemical processes. In order to assess the quality of the protein in our nutrition, scientists have developed a variety of methods and standards to describe proteins and peptides in their qualitative and quantitative composition, as well as their attributes regarding their behavior in our body in their pharmacological ADME profile. ADME is the abbreviation for absorption, distribution, metabolism, and excretion. For the protein quality assessment, the factors of qualitative composition, digestibility, and bioavailability are crucial for the evaluation. There are many measurement techniques and scales for protein analysis available. Among the most frequently used measurements and scales are the following 4 methods. 1. Net protein utilization. 2. Protein efficiency ratio. 3. Biologic value. 4. Protein digestibility corrected amino acid score. In my next blog, I will describe these methods and their relevance for the quality assessment for proteins in detail.